Wobble hypothesis, which is also known as Wobble pairing, is related to the field of molecular biology. See, at the time of translation, that is protein synthesis, the transfer RNA carrying an amino acid will be coming to the messenger RNA. It will pair with a particular codon of messenger RNA. Actually, the anticodon site of transfer RNA would be pairing with the codon part of messenger RNA. But at that time, what we observe that the third letter of a codon will have very flexible pairing with the first letter of transfer RNA. So we know that there is a strict pairing between AU, A being a purine base that pairs with U, that is a pyrimidine base. Likewise, G pairs with C. So there is AU and GC pairing. Such pairing is Watson-Crick pairing. But this strict pairing will not be followed at the third position of a codon and the first position of the anticodon. So let us see the details of this aspect. See, the Francis Crick was the person who for the first time proposed the Wobble hypothesis, and he proposed this hypothesis in 1966. Actually, the current estimate indicate that there are 30 to 40 transfer RNA types or species which are present in the bacteria, and up to 50 transfer RNA species are present in animal and plant cells. So we know that there are 61 codons which actually encode for specific amino acids. But there are quite less number of types of, not number, other types of transfer RNA in case of bacteria as well as in eukaryotic system like animals and plants. So it means Wobble pairing that will definitely be allowing a single transfer RNA to recognize or to identify more than one codon. So this is the main point one should understand, that because of this Wobble pairing, that is flexible pairing occurring between the third letter of codon and first letter of anticodon, the number of transfer RNA in the cytosol, not number. I mean, when I say number, I actually mean to say the types, means different types of transfer RNA. Their types gets reduced. So you can have better idea by observing this diagram. What is shown in this diagram, the transfer RNA is this one. It is carrying amino acid with it. And here on this end you can see there are three letters or three nucleotides which form the anticodon. So this is first nucleotide because it is at the site of 5' end. So this is first nucleotide. This one is second and this is third. So these are three letters, first, second and third. Whereas if you observe the messenger RNA, then in this codon, say this is a codon and this one is first letter, this is second letter and this is third letter. So flexible pairing will always be taking place between the third letter of messenger RNA and first letter of transfer RNA. Whereas the other two, the first letter of messenger RNA and third letter of this transfer RNA will have the Watson-Crick, pairing rule. That is, here if A is present over here, it will be pairing with U. In the second position, if G is there, it will strictly be pairing with C. But there would be flexibility only at the third position of this messenger RNA. So this is Wobble position. Okay. Now in this figure, you can see that this is normal situation. In the first case, this one is normal situation. What is shown here? that this transfer RNA is carrying leucine. Leucine is attached with its 3' end, and at its 5' end it is G here. So this is first letter G, and in normal situation it has to pair with C, that is third letter of messenger RNA. So the codon is CUC, and it will be pairing with anticodon GAG. But because of this wobble pairing, what is happening here, what is shown here, that the G of the first letter, means first letter G of anticodon, instead of pairing with C, it is pairing with U. So the U is another pyrimidine base. So this kind of flexible pairing could be there. Means if it is C over here, it will definitely be pairing with that. But even if it is U, that is uracil, then the pairing will take place over here. So this is wobble pairing. And this is a table through which we can have idea that what sort of flexible pairing is possible between the first letter of transfer RNA and the third letter of messenger RNA. So base at first position, that is 5' end of transfer RNA, and base at third position of messenger RNA. So if it is A at the fifth position, or at the 5' end, not fifth position, at the first position of transfer RNA, that it will be pairing with U. Likewise, if C is there in anticodon as first letter, it will be pairing with G. And if G is there as first letter in transfer RNA, it will either be pairing with C, because pairing with C is a usual kind of pairing, but it may also pair with uracil, that is U. So this is wobble pairing. Likewise, if uracil is there as first letter in transfer RNA, then it will be pairing with A, which is actually normal pairing, but it can also pair with G of the messenger RNA situated at the third position. There is one more, kind of base, that is inosine. It is a different form of actually guanine. So this inosine, it has ability to pair with A, it can also pair with U, and it can also pair with C. So if at first position in transfer RNA I base is there, then it has ability to pair with 3 bases of the third letter of messenger RNA, that is A, U or C. Except G, it will be pairing with other 3. Here exactly same diagram is there, that is this is transfer RNA, and at its fifth position, fifth means 5' end, or you can say the first letter is G. This G has ability to pair with C, that is third letter of messenger RNA, because this lower line is of messenger RNA. This is a codon UCC. If this codon is there, then the anticodon GGA will be used for pairing purpose. But even if it is UCU, then same transfer RNA will be used. So this is wobble pairing, and that is why in this table you are observing that G present at the first position in transfer RNA is efficient enough to pair with cytosine, or it can also pair with uracil. So this is flexible pairing between the third letter of codon and the first letter of anticodon. Now let us see significance of this wobble pairing. As I just said, that there could be quite less different types of transfer RNA than the number of codon. We know 61 codons are there, so we expect that 61 different types of transfer RNA will be required, but this is not the situation. So cells, because of this wobble pairing, cells have limited amount of transfer RNAs. Thus wobble pairing allows for broader specificity. The thermodynamic stability of wobble base pair is comparable to that of Watson-Crick base pair. This is another important thing, and wobble pairing allows faster dissociation of transfer RNA from messenger RNA. that is at the time of protein synthesis, the transfer RNA bringing amino acid will be pairing with the codon, but because of this wobble pairing, the dissociation event will be faster. See, this wobble pairing minimizes the damage that can be caused by misreading of the code. For example, if leucine, see, we know that leucine that is encoded by the codon CUC, and the other three codons like CUC, CUA and CUG also recognize leucine amino acid. So when you will observe the genetic code dictionary, you will find that there are actually six different codons which identify leucine or which actually encode for leucine amino acid. So if it is CUU, then it will be identifying leucine. But because if it is being misread as CUC or CUA or CUG during transcription of messenger RNA, the codon would still be translated as leucine during protein synthesis. So even in the change in the third letter, because you see the first two letters are same, CU is here, CU is here, CU is here. So in all these four cases, the first and second letters are same. Only change is there in the third letter. So even if change in the third letter is there, that is not going to influence the type of amino acid coming over there. So this is significance of this wobble pairing.